Hey, Aaron, before too many people come, I just wanted to say thank you for this. You know, it's been a great 3-year run here. And I know everybody in San Diego really appreciate it. I'm sorry, I was away giving the talk when you visited, sorry. But thank you so much. I think you did a fantastic job. I know how much work it is having organized a series myself. Thank you, thank you, thank you. Okay.
Thanks, Stuart. Sure. Good to see you. Hey, Jiao.
Hey, how's it going?
Good.
Nice hoodie.
Yeah. Felt weird putting it on for one last time.
Well, you can wear it afterwards unless you want to burn it as a symbol of something.
I haven't thought of it that way, but that could be good.
Or you can frame it like those Steph Curry jerseys and stuff like that, right?
Something like that. You're in your crash pad in
I'm in my crash pad, yeah. Got in this afternoon. I had a nice weekend in New York.
I'm gonna be up at Berkeley on Saturday night.
Okay, what's going on?
There's some party at one of your colleagues' house.
Okay. Should I crash it?
Definitely. Yeah, no, especially as the brain director.
You can DM me whose house it is if you don't want to announce here.
You could probably figure it out, but yeah, it should be good. But then Sunday morning, I'm going to Baltimore.
Well, let me know if you want to come up a little early and hang out on Saturday. I'll get back. I should be back by around noon or so.
I arrive from Shanghai at like 3:30 p.m.
Oh, you're flying in from Shanghai?
Yeah.
All right. Well, let me know if you want to hang out or if you want to crash in my house after.
Cool. Yeah. It's sort of like a party / discussion about AI. I'm going more for the party thing, but the AI thing could be interesting.
Okay. I see Mosca.
Oh yeah. He's a
I
stalwart.
came prepared today.
Oh my God.
I figured this was the only appropriate thing I could do for a last
Holy
day.
shit, man.
I know that's not even a filter.
It is not a filter. Although
Are you here?
I was playing with backgrounds earlier and I had the Flybrain or Twin Peaks which this is the most hilarious thing I think I've ever seen.
Wow.
But I thought the very least I could do would be to dress up.
Wow.
Well, Aaron and I did have this discussion earlier, but he opted for the hoodie. Like Don Lemon, as it turns out.
That's fair. You know, both my hoodies are in my office and I am not. Otherwise, I might have done the hoodie, but I figured, you know, do this properly, do it respectfully.
Yeah, I hadn't thought of it. My hoodie's 3,000 miles away, so.
Oh yeah, I should bring you your new hoodie, John.
Yeah. Well, how long are you going to be in town for? Hey, you mean?
Hey, John.
It's on, right?
Hey, you're from Tokyo.
Yeah.
Oh, whoa.
How's it going?
You're supposed
That's
to be
awesome.
on. Did he
Yeah.
hear me?
I think
Thanks,
so.
Mommy.
John, why you're not here?
I know. Sorry, I'm not there because I'm here. Bethesda. Sorry.
It's very metaphysical.
Yeah. I'm with you in spirit.
What is it?
Yeah.
I
Oh,
see.
sorry.
Yeah,
Where are you guys? You look like you're in somewhere really fancy.
oh, yeah. Yes.
Yeah. But it looks
God,
like
look at
it's
those chairs.
there. But it's not night. It's 10 a.m. in Tokyo.
Yeah, where are you guys?
We're at the hotel lobby.
But it looks dark.
Yeah. It's a little bit dark, yeah. But
Hmm.
this is their hotel in Shinjuku.
The Godzilla Hotel.
Hmm?
Sorry. No. Yeah, we're Tomomi's meeting is starting very soon, so we wanted to see the beginning of people signing up.
Are you going to head off to the meeting?
Yeah, everybody wear masks here. So
Yeah.
Oh,
nice.
Jin's got hers on. Nice. I at least have the right color anyway. I'm close.
I have one hoodies at home, one hoodies in the office. So
Jin has 2 hoodies.
yeah, I do. I'm
You're special. You're special. You're special.
Yeah.
Is that a rental, Tim?
No, it's not.
Okay. All right.
Although I probably would have done that if I didn't have one. I would have committed to the bit. But no, this is mine.
Wow.
You're looking pretty spiffy, man.
Thank
Yeah,
you.
great.
The very least in respect that I could do.
Now, you don't have to show us, but I have to ask you: are you wearing the trousers?
Absolutely not. I'm wearing my running shorts. I'm sitting in my office at home. So
you ruined it. You just ruined it.
This is why I'm
You
not
could've-
getting up.
You could have just told us you were.
I'm an honest
maybe-
man, John.
Need to use the password. This is the first time you're setting up password, right?
No.
That's all.
Wow.
It varies.
Hey,
Depends on
Joe.
where you are.
So I expecting some formal dress, but didn't imagine that formal.
Felt
Is
respect.
Zilong wearing the hoodie? We can't
Yeah.
see.
Yeah,
Are
of
you
course.
wearing the
I'm
hoodie?
making like I'm a headphone, so I could see my. It's like here. Wow. So
With Jana, you're DC, is that? Oh,
Yeah, I'm a DC. I'm a DC this week. Yeah.
Mm.
hang on, let me share my screen to set my
Okay.
my new Mac. I haven't shared my screen yet, so I guess I need to come
Oh.
up and we log in again. Yeah, just like just okay. Yeah, I get it exist to come back again.
Aaron, how many Neuro Zooms have there been? 150?
There have been
Close, right?
268 talks,
So divided by 2.
134.
All right.
This has outlasted entire nation's response to the COVID-19 pandemic.
Indeed.
You mean, where are you? The hotel?
You're in Tokyo.
Oh, sweet. Sweet. Oh. Yeah, some friend of mine are trying to have some Neuro Zoom 2.0 version, and we're thinking about how to do it in post-COVID time.
Yeah, just do it.
Maybe not so frequently, but online talks are quite still easier than travel.
But travel is more fun.
Oh yeah. Let me see. I should. It's working, right? Okay. Right.
So Aaron, who's going to introduce you?
No one needs to introduce me.
Well,
Everyone-
we'll have to prepare, right?
I'll have to figure out what to do every Monday at 5 p.m. or Tuesday at 9 a.m. China.
Yeah. In summertime is fine because it's like 8 to 9 p.m. a.m. While early is on. So yeah, we should find out. Also, some European colleagues are trying to have some Neuro Zoom, including Europe. That will be to another time zone. So
Yeah, that's great. Hey, Splinter.
Look what I'm wearing.
Oh, nice.
You see it?
Yeah, looks good.
Oh, you got the new one.
Say it again.
Yeah,
you got the new
that's
one.
Yeah, but that's the 2.0 version.
Is it?
Oh yeah.
Thank you.
Tim, I think you win the best dressed award.
Yeah.
Second place, Shanaz.
The first and only time.
Well,
So who's introducing you, Aaron?
I just said that.
No introduction.
You are Subhajit. It's you didn't get the memo.
I have only like anecdotes. I don't have anything professional that I can.
Well, that's the whole point, isn't it?
No, the anecdotes are a little bit dangerous.
Tim, are you dressed up just for this occasion, or are you off to something exotic?
This absolutely felt like the respectful thing to do, to see it out in style. So as John made me reveal, I'm even like wearing my running shorts right now. So, I've only got the half suit,
But
just enough for the screen.
But wait a second, Tim, you're dressed up for the after party, right?
Clearly, obviously, you're all coming to this, right? I mean, it's Philly, it's 9 o'clock, it'll be great.
I'm gonna hop on the train. I'll be down there in an hour, 2 hours.
Be fine.
Shouldn't there be an award for the most attendance? Who attended the most? Aaron and Zilong. Shouldn't there be an award for that?
Yeah.
Yeah, that's a good idea. Tim is in the
Yeah,
running
from
for-
stress. We are curious if who's running in the first place. But,
Best dressed is one thing, but I assume you have been keeping attendance these years and so on.
Yes, I get a report every week about who attends. I keep note of it.
Well, I hope I'm in the running.
Yes, you're up there too.
I know we have talks today, but I think we all just want to say what an incredible gift this has been and how amazing it has been to, particularly during the difficult times, to have, something stable and to take one's mind off catastrophe and to have hope about humanity and science. So before the talks begin, at least I just want to say that how grateful I am personally for what you guys have done and the learning, and it's been just really amazing.
thank you,
Thanks so much.
I never got to give the talk I was slowly getting ready to suggest giving because of being so slow. So if there is a 2.0 Zilong, you have to send us all
Sure.
emails.
Sure. I'll talk about that. I'll try to including a European colleague, maybe.
Maybe should we get started, Zilong?
Yeah, sure.
Okay, yeah. So thanks everyone for coming. It felt a little bit weird putting on the Neuro Zoom hoodie for one last time, but it's been great seeing everyone here over the years. So today's the final edition of Neuro Zoom. We've been doing this every week for nearly 3 years. It started right at the beginning of the pandemic, and we've had a total of 268 science talks. So just briefly, in looking back and reflecting on the broader significance of this kind of trajectory, I just thought of a few things, a few brief lessons, five quick things. First, it's been an honor to get to virtually sit next to all of you every week for almost 3 years. Second, when Zilong and I first conceived of an idea to do something like this, we never imagined it would last so long or that it would be so much fun. In reality, we just did it because we were bored at home, couldn't get into the lab. We wanted to hear some interesting science. I used my Stanford Zoom account. We set up a meeting in the afternoon, California time, morning China time, and thought we would just talk to each other, maybe our labs. And we sent around the Zoom link to some of our friends and said they could join, but it turned out that they sent it to their friends and so on. So when I logged in, there were 500 people waiting there. That's the limit for the Stanford Zoom room. So then we just asked a couple more of our colleagues to give a talk the next week, and 500 people. And then we set up a YouTube channels so that we could live stream the talks for people who couldn't make it. And many of the talks are archived at the Neuro Zoom YouTube channel. So we all, the third lesson, we all learned that being physically and socially distanced is hard. And we have relearned that being together with each other is sacred. So these weekly 1-hour Neuro Zoom meetings have certainly been sacrosanct to me. And it's really been wonderful to see all of you here week after week for almost 3 years. 4, science is and ought to be an international endeavor and open to all. The importance of scientific exchange is as important now as it's ever been. And I think that when I say we, I don't just mean me and Zilong, I mean all of us. I think we've done a small part in showcasing the best neuroscience in the world, in facilitating new collaborations between laboratories across continents. And all we did was set up a Zoom room and turn on our camera every week. You're really the ones that made it what it is. And 5th, this is the last one. I guess all of the lessons seem obvious when looking back, but I think it's important to attend talks outside your direct field. Sometimes we get into this habit of just going to meetings and attending seminars for topics directly related to our specific narrow field. Zilong and I intentionally mixed up the topics each week because we wanted to hear new things and also to learn something new. I've learned so much over the last 3 years, and I wouldn't have done it any different way if I had to do it again. So thanks so much to everyone for making these past 3 years so much fun. I'm really going to miss this and miss all of you. But reach out. I'd love to hear from you when you're in California or Shanghai or anywhere. And with that, Zilong, I would love to. Zilong has been an amazing colleague and friend, and it's just been great to be able to do this with him. He's now recently moved to be a professor at the Shanghai Jiao University, and he'll talk to us about that and his newest research. And take it away, Zilong.
Sure. Thanks so much, Aaron. So maybe just spend 1 minute before my talk to express my gratitude to all of you. So first, I don't have so many great talks that Aaron can give the speech as Aaron did. So let me just say a few thanks. So first, thank all the audience. So every week, it's been a quite emotional time. So I can enjoy science and see a lot of old friends, new friends to enjoy the every different topics every week. So so everything has beginning, has end, but it's quite a memorable experience. So second thanks I would give to Aaron. So Aaron did like 90% of the work. So finding speakers and setting up the posters, and all posters done by Aaron himself. So it's been quite a gallery. So on the neurozoom.bio website. And also I want to thank a lot of audience, especially in China, is our graduate students. I know they have a kind of weekly seminars, online seminar series every 8 a.m., 9 a.m. They set up the screen to listen to talks far away. So so we're trying to set up some Neurozoom second version. So try to make the tradition keep going. But also, what we will like more is try to meet each other offline in the international meetings in China, in US, in Europe, in the workplace. I think all these academic communication is about. All right, so I see all of you, so many familiar friends. I think one besides thank you. Also, I want to share with you the latest immature neurocircuitry work from my own lab. Okay, right, let me get started. So, okay. See the screen, yeah? Let me see.
Starting to share.
Did I?
Yeah, looks good.
All right. So the work is the first really the circuitry work from my lab. So I'm trained as a biochemist. So beside the neuro development, I've always dreaming to find out what the circuitry control the social behavior. So the title is inhibitory input from hippocampus to ritual splinter cortex. So I'm recently moving from ION to Shanghai Jiao University. So I don't have spent much time to talk about autism. I'm sure all of you are familiar with. It's a focus in my own lab for almost over a decade. So it has many core symptoms like defect in social communications, as well as the repetitive stereotypic behaviors. Now, in my own lab, recently, I'm thinking we're trying to address this complicated disorder from 3 dimensions. The first is causes. By talking about causes, we mainly focusing on the genetic causes. We are finishing the first large Chinese ASD cohort. I'll talk about that in a minute. So the second is trying to the mechanisms. We really try to finding out what the neurocircuitry, so controlling the social behaviors in mammals and including human. And third place, we're trying to use the including gene therapy, neuromodulation, also some pharmacology method trying to see whether it's possible to have some therapeutic methods to address the autism spectrum disorder. Now, first, let me briefly talk about genetics. So everything is beginning from genetics. So in the 2015, I started collaborating with local physicians. Dr. Du Yazong is one of the major child psychiatrists. In the first work we published last year, actually, so we collected over 300 ASD trials and performing whole exome sequencing for all of them. So the first work, we identified numerous de novo mutations, but the cohort is quite small. So in second cohorts, we starting from 2017. So we work with the Dr. Li Fei in Xinhua Hospital in Shanghai. So by combining two cohorts, we have the over 1,000 ASD trials in China. This is by far the largest ASD trials in East Asia, as I know. So in Dr. Li Fei's work, she also figured out the interesting subtype of autism, which is called high functioning autism. So in this group, she identified this group has a normal IQ and DQ, means they have a normal cognition abilities, but also they have a severe defect in social communication scores. Okay, so by focusing on the high functioning autism, we're trying to figure out which genetic causes as well as the neurocircuitry might dominate social behavior, but not affecting the cognition abilities. This work done by one of my students, Jing Cheng. So briefly, this one data I want to share with you from genetic study. From the 1,141 SC trials, we'll be able to identify 22 genes coming up from the FDR.1 threshold, which is normally adapted by the colleagues, international colleagues. From this 22 genes, surprisingly, there are 9 of them had not been recorded by Safari or US, European, any other databases. So basically, they are highly frequently in China, but not in other geographically populations. This suggests us that we need to performing the ASD whole exome trials in different geographic populations. We might find some new genes. Okay, besides that, we're trying to find out second place, what is the neural mechanism, what lesson could we learn from the genetic studies. So all the neurocircuitry, we're trying to focus on some new targets beside the MECP2 or SHANK3 I already studied, right? So everything is starting from step 1, a potential new high-functioning autism risk genes. So the patients also come from Xinhua cohort. So the kid has a number 113. It's very interesting. He has perfectly normal IQ, say 100, but the ASD score is very severe. Okay, so basically, it's high-functioning autism patients. We validate the mutations from patients, find out it's very severe de novo mutations lead to a premature stop the translation of the protein. Okay, so two of my postdoc Kan and Yuhan carried out the experiment. This work already published. I'm going to tell you the main findings. So what the reason we're focusing on step 1 is that I'm trained a biochemist, so I'm familiar with just the gene for a long time. It is one of the desimulation enzyme. Okay, so simulation is just like phosphorylations and remove the SUMO group from target proteins. So the SAMP1 is one of the enzyme remove the SUMO1 from the target. So it's very critical enzyme, right? So interestingly, one of my colleagues in Shanghai Jiao University, Jin Kecheng, he has studied SAMP1 all his career. He has all the SAMP1, 2, 3 KO and conditional KO mice. So we asked the SAMP1 heterozygous mice from him. So indeed, we find out this SAMP1 heterozygous mice has normal cognition to objects, but defect the social novelty test. And also mice has normal learning memory abilities, suggesting it might be a target for high function autism, only affect the social domain, but not other abilities. So, and one reason we focus, one main finding from this study is that it lead to us to the new brain region, which is the sub-region of the richest cortex. So can I find out is that all over the brain of the SAMP1 heterozygous mice, the one place has some abnormal development features, which is RSA, retrosplenial agranular cortex. So all the places called richest, they are granular region and agranular regions. Okay. Now only on RSA region there is an increasing probability in positive neurons. Okay. So the work already published. So basically, we have no idea what richest might do. There's a review over 10 years. The work by Carl Desimulation in 2020, he find out ketamine can specifically activate neuron in richest cortex. So let us believe it has something to do with social behaviors. Now, briefly, the work already published, you have find out in the richest RSA region, and then sample and heterozygous mice has increasing miniature iPSC, but decreasing miniature EPSC. Okay, now we find out the downstream target or the simulation target of the sample one is FMRP. Okay, this is very traditional, intellectual disability as well as the autism target. We find out the molecular mechanism is a sample one deficiency lead to the over simulation of the FMRP, lead to the degradation of the FMRP. Okay, so work was already done. So we can rescue the social defect using either sample one or FMRP, specifically in the richest cortex. But really, what surprised us is why, right? So why does richest cortex related to autism? So another colleague from Zhejiang University, Xiangyao, find out in Fragile X FMRP knockout mice, there are also some dysregulation neuroactivity in richest granular cortex. Also, we try to find more evidence in the MECP2 transgenic mice, mostly common used autism mouse model. Dr. Zhifeng Liang in ION helped us to perform high resolution MRI, finding out richest cortex, the rehole rate, suggesting the neural activity in the richest cortex, increasing the autism mouse model. And also, mouse is not just enough. So we made the MECP2 transgenic monkey years ago. So Dr. Zheng Wang, now he moved to KU, helped us to performing the functional MRI imaging. So he find out the neural connectivity of the richest cortex in the MECP2 transgenic monkey. Indeed, it's also increased. Another place increase is prefrontal cortex. Okay, so the real tell us richest cortex has something to do with autism, something to do with social behavior. So my postdoc Yuhan set up to find out how the richest particularly RSA, contribute to social behavior. Now, the first experiment Yuhan did is trying to looking at whether the RSA is activated during social interactions. So Yuhan performing the we call it intruder test. The mice is right here, the putting either an object or a stranger mice. The first thing you can find out the neural activity in RSA measured by C-force is significantly increased. Okay, just a few minutes after social interactions. All right, so it seems first experiment work. A second experiment, you can try to use fiber photometry to see whether the real-time neural activity. That's something turns out to be even more interesting. So you can use either excitatory promoter CAMK2 driven GCAM6 or a QI-dependent GCAM6 to inject to the VGAT inhibitory neurons. So what you can find out after social interaction, interesting, is the neural activity come down, right? So see here, after meeting the stranger mice, neural activity is significantly decreased comparing to the mice meeting with the another objects. Okay, so those, what happened to that? Either excitatory or inhibitory all decrease. So it tells us after the first activity increase, there are second decreasing phase. All right. So what happened to it? Then we performing the optogenetic manipulations. Use the two setup. First, we try to consistently activate the RSA neuron using chernodopsin to activate all along. Right. Then we find out we use continuous activation. The social interaction time and the frequency all strongly decrease. That tell us you cannot activate RSA neurons. You will affect the social behaviors. And second, we're trying to activate the RSA neuron right after the social approach shown here. But first, also we're trying to consistently inhibit the activity RSA neurons. Also, they decrease the social interaction. Okay, so you cannot all the time. You cannot all the activate or decrease. But second, we're trying to decrease, just mimicking the natural course, decrease the neural activity right after the social contact. Okay, we'll call the sniffing start inhibition approach. It uses inhibitory rhodopsin, and it shows this approach can increase the social behavior by increasing the cumulative total sleeping time and the sleeping time each sleeping interaction time in every second or a single time. Okay, so this tells us we cannot activate or decrease all the time, but we can increase the social behavior after the after mouse makes the social contact. So what happened to it? Where is the inhibitory input come from? This work has been published by many colleagues. There's a inhibitory input either from VIP neuron in hippocampus or PB neuron from hippocampus. It's very old literature. So first, you can inject the CTB in RSA, find out it's over 40% of the parabolic neuron in the hippocampal CA1 region, basically projecting to there. Okay. So second, you can use another way, inject the crate-dependent GFP and the terdopsin in the PB cream mice and find out there's a synaptic terminal of PB neurons accumulate in the layer 2-3 of RSA. All right, second, you can use the physiology. First, inject the crate-dependent terdopsin in the PB CA1, the PB crate, using the perfectly fast spiking features and using the activated the PB terminal shown here, shedding light to the RSA, and find out there are strong connection from hippocampal CA1 PB neuron to the RSA. It's quite far connections. Or after make the connections, we asking whether the inhibitory input is sufficient to make it the social behavior inhibition. Then we before asking that, we were asking by reviewers to show the PB neuron in CA1 is responsible for the social interaction, right? So here, indeed, we injected crate-dependent GCAM6 to the CA1 and the PB crate to expose either objects or strangers. When exposed to the normal mice, the neural activity of PB in CA1 strongly increase. It's social responsible. Then we use this approach to first manipulate the CA1 PB neuron by injecting either crate-dependent M-cherry or M4D inhibitory pharmacogenetics, or we try to manipulate the neural activity locally in RSA, injecting CAMK2-driven GCAM6 right locally in the RSA. And then we can manipulate the circuitry by locally infused signal. Also, we did the IP inject signal as another control. Their results are consistent. The first is all the down phase things coming perfectly in the control experiment. And second, we were blocking the PB to RSA inhibitory input. The down phase seems to be abolished. Okay, that shows indeed the RSA down phase is contributed by PB CA1 PB. And the second is asking whether the social interaction will be altered. Indeed, in control is fine, but we block this CA1 PB inhibitory input, the social behavior decreased. Okay, so we cannot the inhibitory input is required for the normal social behavior. And also we use the 3-chamber test to show normally the mice had the normal social approach and the social novelties. Now after the blocking the CA1 PB to RSA, all the phases are decreased, are abolished. Okay. Now the one last experiment we did, I tried to asking whether the circuitry is working in the autism mouse models. This way we use another well-established MAP2C mouse model. Also, thanks to the Professor Stuart Lipton. He is a pioneer using the MAP2C to study the mechanisms. We use the mouse from his studies to show its mouse already find out MAP2C is well-established autism genes. So first, we're trying to label the hippocampal CA1 PB to RSA by injecting the AAV PB promoter-driven CHR2 or cherry to here. We can label it mostly 80% of the PB promoter is very visible. Also, we're trying to targeting by show we look shedding light to the RSA we can receive the synaptic input from CA1PV to RSA in the MAP2C heterozygous mice. Right, then we try to manipulate the circuitry in the MAP2C. First, we use the consistent activation, the input all the time, right? So not surprisingly, it decreased the social interaction even in the MAP2C abnormal mice. This mice already has some defect in social interaction. The increasing the circuitry even decreased the social interaction further. The second, one last experiment, we're trying to use the sniffing start inhibition. So after contact, we activate the CA1PV to the RSA inhibitory input, and we can rescue increasing the social interaction in the MF2C heterozygous mice, shown by the total sniffing time and the single sniffing time per second. All right. So this is it for the data. So let me tell you what the model is. So that we don't have a final model. So for now, we have the two-phase social behavior processing hypothesis. So firstly, the spinal cortex, just like many brain regions, that could be activated by social cues. The cues, that's the wrong word. So but interesting is second phase. So we propose the CA1PV inhibitory input might serve as the filter, right? So the way is to block the non-social sensory information, because if you activate all along, you might not able to figure out what is social, what is non-social information. So that might play a key role. For example, in autism, there are many hypotheses. One of them is sensory information defect, right? So some people say it has hypersensitive to the sensory information, but it really didn't tell us why hypersensitive social information might lead to autism, right? So here we propose one of the possible routes is autism is due to the incapable of filtering out non-social sensory information. That might lead to the real social information cannot be stretched out from all the outside informations. Also, as many questions left, so we need to figure out exactly how the social or non-social information is processed within the millennials or the input after output, etc. All right, so it's my time. So I've had another work coming up, and hopefully still working on the gene editing, base editing of the MAP2C. So hopefully it'll come up the next. We're still revising the work, hopefully to share with you in the future. Now I'm moving to the Songjiang Research Institute in the suburban area of Shanghai. So I hope we are making the new building in a couple of years. So soon we have a 10 junior PI in our institute already. So hopefully we can host you to visit Shanghai, visit Songjiang, a beautiful place. It's a new institute. It's all focusing on the neuroscience. We're hoping have a good timing with you in Songjiang. Also, SRI is recruiting. So all of you interested in moving to Shanghai, please talk to us. Talk to Professor Duan. We're happy to take a look. All right, so it's my talk. I'm very happy to take any questions.
So long, that was great. It's Stuart. Thanks for using our model. We have now developed a MEF2C activator drug. You might want to try. I'll write to you.
Sure. Thanks. Yeah, your work is very inspiring.
Remaining a good allele of MEF2C to equal as if it were not haploinsufficient. And so it works in our models. Might be really interesting to try in your model.
Sure.
Email. See the beauty of this? You can't stop these talks.
Thank you.
Thank you. And thank you for organizing too. I had thanked Aaron, and I didn't know if you were on yet, but you guys have both done a great job. Thanks.
Thank you. All right.
Hi.
Hi, Kevin.
Yes. Hi. All right. Okay. First of all, thank you very much for both of your great efforts in contributing all these great seminars and then it's the questions. It's very interesting to know that the the inhibitory networks actually contributes to the social, differentiations. So, but my question is, what do you think it's you're talking about retrosplenic cortex. So you think the deficits in social disconnections comes from the retrosplenic cortex or the hippocampus PV network? So because you're talking about the connections between these two brain regions, right? So in the autisms, I'm pretty sure the inhibitory neurons, both in the hippocampus and as well as in the cortex, contributes to grade of the lack of inhibitions. So what is the role of retrosplenic cortex in your context?
Thank you for the question. So actually in the MAP2C heterozygous mice, the PV population of the PV positive neurons is all decreased. So all over the place, exactly. So all the hippocampus, all the many cortices is all decreased. So, but we are proposing. So although there are, for example, if because the MAP2C has a very strong transcription activator, right? So might regulate the PV development for some reason, or has some neuroactivity dependent PV because the PV itself is rely on neuroactivity. But what we're thinking is maybe the hippocampal C1, PV2, the richest splinter cortex play a key role in the controlling sensory information processing. So, for example, in our model, by manipulating only this circuitry, we'll be able to rescue the social behavior. So, although the PV development is all over the places, but there are some few key circuitry we can manipulate. Well, that contribute to social behavior specifically. That's what we're thinking. Yeah,
that's very cool. And do you know the PV neurons in the PV neurons in the campus also project to other places, right? So in addition to retrosplenic cortex. When you're tracking using the rabies virus.
we're trying working on that, but we don't have much clue yet. So hippocampus VIP neurons also projecting to another layer, deep layer of a splinter cortex. Also, the hippocampus somatic SST neurons has some interesting connections. But PV, we haven't saw many. We find some virtual people published the virtual C1 PV has some interesting real social memory, right? But in dorsal C1 PV, we need to do more work on that. We don't have much clue. Okay,
great. Thank you very much. Great talk. Thanks. Thank you both again.
All right. So
thanks.
No more questions. I need to introduce Aaron. So I need to talk about the first time I met Aaron. It's all in China. It's in Shenzhen. It's 10 over 11 years ago, right? So now we have an Aaron. I didn't imagine I can work with Aaron for 3 years, so organize seminar every week. So as you remember, Aaron did his bachelor degree in Penn State, and the PhD in UPenn, and a postdoc in MIT. And now Aaron started his legendary career in UPenn. I tried to using a very simple system, East screen system, to finding out all the conversional protein aggregation process is being implicated in many neurodegeneration disorders. I still remember, so in the first talk from Aaron, I really opened my eyes how you can use a simple system to address important questions. So after a few years, started amazing work in UPenn, Aaron was recruited to Stanford. So where Aaron started moving his work from east to the high mammals, mouse models. So besides the several amazing work in mouse model, addressing using ASO and other process, Aaron has recently also worked using human samples to tell us more about the ALS and many neurodegeneration disorders. So I think Aaron has many more work to come, has many new surprises to give us, surpassing today. So Aaron will tell us, what is the two ALS drug target, particularly what is the new ALS new drug. Okay, welcome, Aaron.
Thanks, Zilong. Great talk, and thanks everyone for letting me be the last speaker of this series. So I can't help myself being able to organize meetings and things. So I just wanted to advertise that even though our online meeting is done in a couple of weeks, we'll have a great ALS meeting in person in Shanghai. Everyone in China is welcome if you can get to China and come to the meeting. It's completely free. All of the best, every top ALS researcher and clinician in China is going to be there. So please come. It's going to be a really exciting talk. You can scan that QR code. I learned how to make a QR code, and it links to a webpage that you can register. Registration's free. And there'll be other special treats and things like that. Okay. So let me start. Okay, so today I'm going to talk about neurodegenerative disease, and these are different types of diseases, and they have different clinical presentations. Some of them affect memory, some of them affect movement, but there's one theme that unites all of them, and that's protein misfolding. Proteins become unfolded and aggregate and accumulate in the brains of patients with these diseases. And different proteins aggregate in different parts of the brain, and mission of my laboratory is to try to define the cellular pathways that are affected by these misfolded disease proteins. Today, I'm just going to talk about one neurodegenerative disease, amyotrophic lateral sclerosis, or ALS. This is a disease caused by loss of motor neurons from the brain and from the spinal cord. When these motor neurons innervate muscle, and when these die, it causes weakness, muscle weakness, and that progresses to paralysis and then ultimately death. It's a very fast progressing disease. There's no cure. There's 3 FDA-approved drugs that are minimally effective. So the understanding the mechanisms of the disease will hopefully lead to the development of more effective therapies. ALS is also associated with misfolded proteins. The protein that misfolds in ALS is an RNA binding protein called TDP43. It was discovered by my colleagues at the University of Pennsylvania, Drs. John Trojanowski and Virginia Lee. It's an RNA binding protein. It regulates splicing. And then in disease, it gets depleted from the nucleus, and it builds up here in the cytoplasm in these large, round, Lewy body-like or these skein-like aggregates. Shortly after they discovered TDP43 pathology, geneticists discovered that mutations in the gene encoding TDP43 are a cause of some rare forms of sporadic and familial ALS. So that's focused the entire field in ALS on trying to figure out how TDP43 might contribute to the disease. This slide illustrates the question and problem. On the left is a section through a human spinal cord, and you can see a large motor neuron. This is the nucleus. This is the nucleolus. You can see TDP43 is diffusely localized in the nucleus. You can imagine that it's binding to RNA, regulating splicing, doing all its normal functions. And on the right is the disease situation. Now you can see a couple things going on. You can see these large aggregates out here in the cytoplasm, these ropey, skein-like ones. You can see some smaller inclusions of TDP43. So maybe TDP43 is causing disease by aggregating some toxic function in the cytoplasm, interfering with other RNAs or RNA binding proteins. On the other hand, you can see now that TDP43 is depleted from the nucleus. It's not where it's supposed to be, so it can't do what it's normally supposed to do. So maybe disease is caused by a loss of its function from the nucleus. These two mechanisms are not mutually exclusive. They both could be going on at the same time, but we just have to consider both of these. Work in my laboratory over the years has been focused on trying to learn about both of these mechanisms, and we've had some projects trying to figure out mechanisms and modifiers of the cytoplasmic aggregates of TDP43, and a recent study looking at some of the consequences of loss of TDP43 from the nucleus. We started our studies using a very simple model system, as Zilong mentioned, the budding yeast. It's a single cell, no brain, no spinal cord. It divides by budding about every 2 hours. But the key is that all of the main pathways are conserved from yeast to human. And importantly, whereas these diseases take decades in people, I can put some of these human disease proteins into yeast, and in just a couple hours, they'll start aggregating and eliciting phenotypes. And then we can perform screens to look for modifiers of those phenotypes, and then go back and forth between these other model systems. So the first experiment we did was to see if we can use this yeast model to study this ALS protein. Is we just expressed human TDP43 at very high levels in yeast, It formed these cytoplasmic accumulations shown here, and it was very toxic. These are spotting assays where we serially dilute yeast cells 5-fold and put them onto agar plates. They can form colonies if they express GFP, but they're unable to form a colony if they express TDP43. And we have this as an inducible system, so we can turn off TDP43 and switch it on based on the carbon source. So this system is really amenable to doing genetic suppressor enhancer screens. So we have our yeast cells that are about to express TDP43. Can we find genes in the yeast genome that, when upregulated or deleted, can either suppress or enhance the toxicity? And that gives us some clues about what was wrong in the first place. So you can see we can do these screens very in a high throughput manner. We can find genes that, when overexpressed, can allow the cells to grow better or worse. We've done these screens many times. One of our top hits is a gene called PBP1. The name doesn't matter, but we found it as a overexpression enhancer of TDP43 toxicity. Yeast cells grow slow when they express TDP43. They grow even slower when you express PBP1. Importantly, it's specific. PBP1 does not enhance the toxicity of alpha-synuclein, a Parkinson's disease protein, or a mutant fragment of Huntington. Conversely, deleting PBP1 was sufficient to suppress the toxicity of TDP43 in yeast. So this is a dose-sensitive modifier. We focused on PBP1 because it has a human homolog involved in another neurodegenerative disease called spinocerebellar ataxia 2. The gene's called ataxin-2. It's one of these polyglutamine disease genes like Huntington. And we collaborated with my colleague at the University of Pennsylvania, Dr. Nancy Bonini, to test this interaction in Drosophila. And she had made transgenic flies to overexpress or lower levels of ataxin-2. Together with her, we've been doing the same thing with TDP43. TDP43 expression at very moderate levels leads to a mild degenerative phenotype in the fly eye, no effect at all if you express ataxin-2. If you overexpress them together, massively degenerating eye, and then lowering ataxin-2 was sufficient to suppress this mild phenotype. So this was cool because this meant that that genetic interaction we discovered in yeast works the same way in an animal. So we started thinking about the ataxin-2 gene. I said it's one of these poly-Q genes. What that means is there's a CAG repeat in the coding region that encodes the amino acid glutamine, which is abbreviated Q or poly-Q. There's almost always 22 or 23 of these repeats in the normal situation. If it gets expanded to greater than 34, it causes spinocerebellar ataxia 2. But because we found it in a screen for an ALS protein, I was just wondering, what about something in between, longer than normal, but not past that range for SCA2? Could that be associated with ALS? To test that hypothesis, we collected about 1,000 ALS patient genomic DNA samples, about 1,000 matched controls, and just measured the repeat by PCR. And we were lucky. We found that almost 5% of the ALS cases have these intermediate length ataxin-2 poly-Q expansions between 27 and 33. So we propose that these poly-Q expansions are a risk factor for ALS. And this has really set my laboratory on a course to try to figure out, how is ataxin-2 contributing to ALS? What are the effects of these poly-Q expansions in ataxin-2? We have some evidence in the laboratory that these intermediate length repeats increase the stability of ataxin-2. So increased stability of ataxin-2 associated with increased risk for ALS. Yeast and flies, lowering ataxin-2 protects against TDP43. I was thinking maybe ataxin-2 itself could be a therapeutic target. To test that hypothesis, you have to do experiments in mouse. So these are all experiments performed by Lindsay Becker, a graduate student in my laboratory at Stanford, who's now a postdoctoral fellow at Princeton University with Dr. Cliff Brangwyn. So what Lindsay did was she crossed a transgenic mouse that's engineered to express TDP43 throughout the nervous system. This is a very aggressive, fast progressing mouse model. These mice all die, become paralyzed and die of degeneration by 30 days. And then she crossed these to 2 independent lines of ataxin-2 knockout mice. These mice are fertile and viable. And then she analyzed mice with different levels of ataxin-2. The wild type mice expressing the TDP43 transgene died by day 30, but she saw a significant extension in survival in mice that were heterozygous for ataxin-2. And she found this dramatic, unprecedented extension in survival in mice that were completely knocked out for ataxin-2. So we're not going to be able to just knock out a gene in a person yet. Maybe with Zilong's new CRISPR technologies, we will. But for now, we started collaborating with a biotech company, Ionis Pharmaceuticals, who pioneered the use of antisense oligonucleotides. These are DNA-like drugs that bind to an RNA they're designed against. Almost all cells have an enzyme RNase H, which recognizes this DNA-RNA hybrid, catalyzes its destruction. So we injected either a scrambled sequence or an ataxin-2 targeting sequence into the ventricle of a day one TDP-43 transgenic mice, and then analyzed the effects of these treatments. So a single administration at day one was sufficient to lower ataxin-2 RNA and protein, and had this, again, a one-time administration at day one, had a significant extension in survival with some mice living over 150 days, whereas all of the scrambled ASO sequence mice died by day 30. This is anecdotal, but this video shows you how strong the rescue can be. These are 3 TDP-43 transgenic mice at day 20. We injected all 3 of them at day 1. These 2 we injected with scrambled negative control ASO. They're paralyzed. And this mouse, we delivered the ataxin-2 targeting ASO. And this mice goes on and lives much longer and is relatively unimpaired. So we're very excited about the power of the ASOs to rescue these phenotypes. And we're equally excited because I mentioned this at the beginning, TDP-43 pathology really is seen in nearly every ALS case, except rare exceptions like individuals that have SOD1 mutations or FUS mutations. And then importantly, TDP-43 pathology is also a major component of a related disease called frontal temporal dementia. So we hope that targeting this modifier gene may have therapeutic benefit for many ALS patients. We're completely inspired by the development and application of ASOs for the treatments of motor neuron diseases like spinal muscular atrophy, and in clinical trial now for other forms of ALS. And excitingly, Ionis, together with Biogen, have launched a phase 1/2 clinical trial to test ataxin-2 ASOs in ALS patients. We're really cheering them on. We're not working with them on this, but cheering them on from the sidelines, hoping that it works. But meanwhile, we're thinking, could there be another way of lowering ataxin-2? Could we somehow come up with a small molecule that could lower ataxin-2 or some other way? So before we could do that, we need to first figure out how ataxin-2 is regulated. So these are 2 screens from Garam Kim in the lab and Caitlin Rodriguez, and both same idea, but different approaches and really cool results. Excited to share them with you. First, Caitlin. So what Caitlin wanted to do is figure out how ataxin-2 is regulated. And she engineered a human cell line that has to have high bit in frame with ataxin-2 using CRISPR. And high bit is an 11 amino acid fragment of nanoluciferase. It's inert, but then when you add large bit, it complements nanoluciferase, it emits light. So basically we have a cell line that just tells us based on how much light is emitted, how much ataxin-2 is in the cell. It's very sensitive. If we lower levels of ataxin-2, we see a corresponding decrease in luminescence. So Caitlin miniaturized this assay and performed a genome-wide siRNA screen in 384 well plates. She has all the right negative controls and positive controls on board. And again, very simple approach. Just look for other genes that when knocked down lead to the cell having less ataxin-2. After filtering and validation and stuff, her top hit is RTN4R, which is also known as the no-go receptor. This is why genetics is fun, because now we have to work on the no-go receptor. You can see here, lowering ataxin-2, of course, with siRNA lowers ataxin-2, but lowering RTN4R with siRNA also lowers ataxin-2. Here's confirmation with the Western blot. Caitlin figured out that this works at the level of the protein because knocking down RTN4R does not affect ataxin-2 mRNA levels, and blocking the proteasome with MG132 abolishes the ability of RTN4R knockdown to lower ataxin-2. There's a peptide inhibitor of the no-go receptor, NEP1-40. Adding this to cells rapidly lowers ataxin-2 levels. Caitlin next tested this in neurons, first in mouse primary neurons, and then in human iPS-derived neurons. Lowering RTN4R in mouse neurons results in less ataxin-2, as does lowering RTN4R with shRNA in human neurons. Same thing with the peptide inhibitor. Mouse neurons, there's less ataxin-2, same with the human neurons. And then she also analyzed RTN4R knockout animals. And she found that these mice are happy, healthy, viable. They just have less ataxin-2 in their nervous system. So this is exactly what we're looking for. And this is kind of interesting observation. We haven't worked on this, but so RTN4R is well known to all of you, the no-go receptor, because of its role in axon regeneration. So we wanted to see, like, maybe there's a reciprocal relationship. So Caitlin performed axon injury and then regeneration experiments in microfluidic chambers. And indeed, if others have found this previously, she validates this. If you knock down RTN4R, injured axon can regenerate more robustly than a control neuron. But interestingly, if she knocks down ataxin-2, it also has the same effect. So perhaps these work in similar ways in this pathway, and that's yet to be explored. Okay, Garam's screen. Garam also wanted to find regulators of ataxin-2, but she wanted to do it a different way. So she designed a screen to use fluorescence activated cell sorting to detect levels of ataxin-2 with no tags, no nothing. I didn't think this would work because ataxin-2 is not a cell surface protein, but she generated an allelic series of cell lines that have all different amounts of ataxin-2, calibrated her antibodies to work, and then got it so that she could sort them based on levels of ataxin-2. She took this cell line that has Cas9, transduced it with a genome-wide library of guide RNAs, 10 guide RNAs per gene, 20,000 genes, 10,000 negative control guide RNAs, all pulled together, all barcoded, stained the cells with ataxin-2 antibodies, sorted them into ones that have low or a high ataxin-2, isolated the DNA, and then counted the barcodes. You can see some of her validations. She has some of these knockouts lead to cells having just as little ataxin-2 in them as if you were knocking out ataxin-2. And then, but her top hit were all subunits of genes encoding subunits of this lysosomal vacuolar ATPase. Whenever you do a screen and you get every single subunit of a complex, it tells you there's something about that complex for the biology you're studying. You can see she made individual knockouts of subunits, and they just have less ataxin-2. This effect is specific to ataxin-2 because knocking down this or inhibiting this enzyme does not affect levels of Huntingtin or ataxin-1. It seems specific to ataxin-2. The other reason that Garam became excited about this is because it's already the target of a known FDA-approved drug called etidronate. And it's been approved by the FDA almost 50 years ago and used to treat a bone disease, osteoporosis, and another bone disease. So we wondered whether this drug could also lower ataxin-2 levels. So we got this drug in, and indeed, it's incredibly safe. I mean, that's the advantage of using an FDA-approved drug. There's decades, literally decades of real-world data on its use, and it's safe over a wide range of concentrations. This is in vitro, this is in neurons, this is 3 concentrations, but shown in the immunoblot, but shown here are more. They just have less ataxin-2. This is mouse neurons, same deal with human iPS-derived neurons, incredibly safe. They just have less ataxin-2. And then she just put this in the water of mice. This is just wild-type mice, or then she put it into this gel-like food that they could eat. And just 1 week treatment, the mice have a little bit less ataxin-2 in their brain. So we wanted to see if this could rescue phenotypes in a TDP-43 mouse model. We can't use that one I showed you the video on because those mice become sick and die before day 30, before the mice can drink on their own. So we used a different mouse model that's developed in Don Cleveland's laboratory. And this one is slower progressing, but it has very reproducible motor impairments. And I actually didn't think that this experiment would work for a number of reasons. So we collaborated with Jackson Labs, and we paid them a lot of money to do this experiment for us because I wanted to save Garam's time because it wasn't going to work. And also they were completely unbiased. So the way they designed the experiment is they took 10 mice that were transgenic for the TB43 or 20 mice, 10 that they randomized to get the vehicle and 10 that they randomized to get the drug. And then they also had other mice, wild-type mice that were given the drug. They baselined the mice with different phenotypic measures. This one I'm showing you is called CMAP, or compound muscle action potential. It's where they stimulate stimulate a muscle or stimulate a nerve and record from the muscle. And it's a readout of the number of axons that are functional and sort of a readout of motor function. It's very unbiased. And so they haven't given the drug yet. They've just aged the mice randomize them, baseline them. So already they're starting to be sick. And then at 14 weeks, they added the drug to the water. And then at 17 weeks, they see that the mice treated with the drug improve. They repeated this experiment because they've never seen mice get better in this measure, but it's true. And then by 6 months, the effect is petering out a little bit, but it's still there. So now we have to spend more money and do this trial again in mice. We didn't realize when we set up the protocol that the mice would already be impaired by 12 weeks. So we're going to start it. So I think it's even more compelling of a rescue if you can reverse the phenotype of a mouse that's already sick. But we're starting again another trial where we're giving the drug earlier, and we're also doing other measurements. Atidronate is in a family of molecules called bisphosphonates. Another one that you might have heard of is called alendronic acid. It's marketed by Merck as Fosamax. And there are other ones I'm not showing you on this slide that our chemistry colleagues have, novel versions of these that our chemistry colleagues have synthesized. So Garam's off to the Broad Institute to do a postdoctoral fellowship. So we're really excited about recruiting students and postdocs to try to figure out how these compounds are working. The ASOs are completely revolutionary, but they cost patients and their families like hundreds of thousands of dollars per year. We bought a gram of atidronate for like $50. So we're hoping that other approaches can also make treatments more broadly applicable. We're also really excited about this type of platform in looking for, doing screens, it's very simple. It's just looking for things that increase or decrease disease genes. For neuropsychiatric diseases, you might want to lower levels or increase levels of haploinsufficient genes. So we're really excited about that. Last slide, it's really great to work with the wonderful collaborators, literally all over the world. It's been great meeting new friends and colleagues and collaborators in China and collaborating with old ones as well. Okay, that's it. Thanks so much.
Oh, what a wonderful talk. Amazing.
Wow, Aaron, a quick question. Amazing talk. Your talk was also amazing, Zilong. But Aaron, Mike, first of all, thank you both for doing this, and I really appreciate it, as everybody else. The other question that I have, Aaron, was with TDP-43. Like in the yeast, when you express a nuclein, you get these vesicular accumulations. I was wondering if with TDP-43, are those really TDP-43 fibrils, or have you done EMs on them, as anybody?
The ones in yeast?
Yeah, in the yeast.
Yeah, so we actually think those green dots you see in yeast are more like a liquid phase of like an RNA protein granule. We've done EMs. We don't have evidence that they're fibrils at all in
really?
yeast.
So it's like just a granular mix.
Yeah, but I bet you, I bet you these ones here, these kind of little teeny ones, they might be more liquid-like, and these ones might be more fibrillar. There's actually some data emerging in the field that, there's data emerging in the field that the fibrillar stuff in FTD might actually be a different protein called TMA106B.
Yeah. I see a lot of hands, but just a quick question about the polyQ expansion. So is it known that the human polyQ expansion leads to an overactive ataxin, or how does that work?
That's our hypothesis, but with some data, it leads to increased stability. We don't fully know the function of ataxin 2, so it's hard to have that readout, but that's our hypothesis.
Got it. Thank you so much.
Aaron?
Hi.
Hi, can you hear me? This is Janine. I also want to thank you very much for your tremendous effort over 3 years organizing the Neuro Zoom. It's oftentimes it's a savior to our sanity when the COVID-19 is the most devastating period of time in China. I have a question. Your work is really interesting. Actually, my question is with your suggestion that with TDP43 ALS mutation, there's a gain of function of ataxin 2, right? Which can be alleviated with a knockdown of ataxin 2, right?
So TDP. So I think the best data so far are that the TDP43 mutations cause a loss of function, loss of splicing function. I think
loss of splicing function. What is the cellular consequence of ataxin 2?
So okay. So aside from TDP43, so just focusing on ataxin 2, we think that the intermediate length CAG repeat expansions in ataxin 2 increase its stability and increase its function. It plays a role in assembling RNA protein granules. We see the altered properties of those. We haven't proven that, because we don't fully know its function. We haven't proven that it's a strict gain of function, but the human genetics and some of our evidence of beneficial effects of lowering it suggests it's a gain of function.
That's right. So it's definitely a gain of function. Otherwise, you won't be able to correct it with a knockdown, right? So the gain of function, what is the cellular consequence of this gain of function? Is neuronal cell death, is neurodysfunction, or inflammation, etc.?
I see. So for sure, in a whole organism, like a person, for sure, inflammation occurs. We're still looking at the molecular level of what are the targets of ataxin 2. It is itself an RNA binding protein. What targets are dysregulated or changed? But we don't fully have a handle on the function to really say. So
in Don Cleveland's mouse model of ALS, presumably you have this gain of function of ataxin 2, right?
Okay, so I don't have evidence that when TDP43 is dysfunctional, ataxin 2 functions increased. That we don't know that. We just know that
not necessarily increase gain of function. It can be a normal function or normal function.
It's possible that lowering ataxin 2 does something else to just blunt the effects of TDP43 aggregation or accumulation or overexpression. It doesn't have to be that the TDP43 is what causes a ataxin 2 gain of function.
Okay.
We don't have any evidence yet, actually, although that is a very interesting hypothesis that TDP43 activates ataxin 2 function.
Well, it should be either gain of function or increase function, because otherwise you won't be able to suppress it by knockdown, right?
Unless, so one model is that TDP43 in an overexpression situation causes trouble by engaging in RNA protein granule assemblies. Ataxin 2 is important for building those. And if you remove ataxin 2, you reduce the propensity of a TDP43 to engage with those. So that's, in a way, it's like independent of a TDP43's effect on ataxin 2.
Okay, all right, thank you. Thank you again.
Any more questions?
Yeah, I have a question.
Holly, right?
And this is Holly Klein.
Hey,
Yeah. Matt Dalva and I basically have the same question, and that is that many patients, at least in the US, have been taking Fosamax, and I'm wondering, or we are both wondering, if there's any information that you can get out of this patient population with respect to rates of progression of ALS.
Yeah, that's a great question. And so it's one we're trying to do. It's a little bit, so there's a couple confounds. So we're doing that. And the one confound with that is mostly women take Fosamax, and there's a slightly decreased incidence of ALS in women. So if you do that analysis, you might get tricked in thinking that Fosamax is protecting. There's all these sort of confounds, but we're doing that. It's a little bit tricky because ALS is rare, but I think that your question is the right question to ask. But at the end of the day, what we really want to do once we do more of the animal studies is just test this in human. Just do a clinical trial, see if it does lower ataxin-2 in human, see if it does improve. But we're now with the medical records integrated, that's the right question to ask.
Vicky, you're muted. You're muted. You're muted. You're muted.
Okay.
Yeah.
Now
Go
is
ahead.
it good now?
Yeah.
Okay.
Please.
So, hi, Aaron. I have a question about the maybe molecular mechanism aspects. Because in your yeast model, you showed that you probably introduced the human disease TDP43 and see the aggregates or the norm, I assume. Did you see did you see that the just like in the disease neurons that the TDP43 gets out from the nucleus to the cytosol? Because I wanted to know that, because I just want to know what you think about this. Because whether going out from the nucleus to cytosol, this process is important for the disease or just a result of the disease. So did you see similar things in the yeast, or that's not what you care? So that's something I'm wondering.
Got it. Okay, great question, Vicky. Yes. So first of all, yeast, they don't have a TDP43, so we overexpress the human one. So they didn't have something to like go from nucleus to cytoplasm. So it's just a model of overexpressing it at very high levels, and it aggregates in the cytoplasm. But
it didn't start in the nucleus.
No, if you express it, indeed, if you do express it at very low levels, I don't think we ever published that. If you express it at very low levels, it does start in the nucleus. But I actually think that one of the main drivers of disease is its loss of function from the nucleus. And there's also important components of the cytoplasmic accumulation and aggregation, but I think it's loss of function first in the nucleus, and then when it gets depleted, of course, more loss of function.
Okay, I see. Also for the ataxin-2, because you use the ASO to knock down the ataxin-2. So, but then does ataxin-2 has a normal function because you knock it down? And also in your patient study, I think not many patients show, not a high percentage of patients show having the intermediate repeats. They seem like most of them still have normal repeats anyways. So because you can see like they probably, most of them still have the normal ataxin-2, but you still knock it down and you have the effect. Can I?
Yeah.
Does it?
Right. Right. Yeah. So okay. So a few things. If you knock out ataxin-2 in mouse, the mice are completely fine. They develop obesity. There's a backup gene called ataxin-2-like. I think that's compensating for it. We've never published it. We made a mouse knockout of it. Those mice die as early embryos. You are right that I think 5% is high, but okay, it is. Or other people could think it's low.
But you helped in all of them, not just 5% of them.
Right. Well, we're not helping people yet, but we hope so. But importantly, the mouse ataxin-2 gene has no polyQ. So we think the polyQ is just a way of changing levels. So we think lowering ataxin-2 could be relevant to sporadic ALS because that is involving TDP43, and it's just the ataxin-2 expansion is sort of an example of its connection to TDP43.
I see. Thank you very much.
Okay, thanks.
All right, Hongyan, question?
Yeah, amazing talk, Aaron. So I'm wondering whether, the question is more about the mechanisms of the rescue when you lower down ataxin-2 in the TDP43 background. So, because as you showed that ataxin-2 also has a novel function in axon regeneration. So lowering that helps to boost up the axon regeneration. Do you think the rescue could also be related with the function of higher axon regeneration in under the stressed condition?
That's a great idea. We I hadn't thought about that. So your point is absolutely correct. We don't yet know what the why lowering ataxin-2 rescues in these different model systems. So we're trying to figure that out. The regeneration part is interesting. We haven't explored that.
Do you know about the TDP-43, whether it's also involved in axon regeneration?
We don't know, but one of its splicing targets is a gene in human, at least, is a gene called STAthmin2, and STAthmin2 definitely plays a role in axon regrowth after injury. So I don't know if ataxin-2 fits in there, but TDP-43 certainly plays a role in that.
Thank you, Aaron. Thank you so much for organizing Neuro Zoom. Yeah, definitely would miss it.
Next question, Yang, please.
Hi, hey, Aaron. First, I would like to thank for the great lectures. And the second, I have got a question about from the clinical perspective. You're talking about many ways to lower ataxin-2, but do you have any idea or clues that there are different populations that may be responsive to the different ways of lowering ataxin-2? I mean, the selection of population of patients.
Great. So this is just what I heard that Biogen is doing. So this is they obviously think a lot about this. So they're in their clinical trial, they're recruiting all ALS patients, I think except ones that have SOD1 mutation. And then they're also trying to have a subset of ones that have an ataxin-2 expansion of greater than or equal to a certain length. And but they're doing that so then they can analyze the effects of the test, whether it works just on individuals with ataxin-2 expansions or on all comers.
Okay, so it depends on the ataxin-2 repeat.
We don't know, but if you had to, I guess I don't know. I was about to say, if I had to think where the best chance of it working would be individuals that have these repeats, but we don't know. I know that the enrolling patients is a little bit slow going, so it would be great if clinicians in China and other countries could also help find some patients. I know that there are a lot of ataxin-2 expansion carriers in China.
Yeah, thank you.
Right. And one last question for Aaron. One last question for Neuro Zoom.
Hi, this is Ruby.
Hi,
yeah, Aaron, really great talk, and Zilong, thank you for your sharing. I mean, you guys are really amazing for 3 years. Like, I really appreciate your coordination, and I think I appreciate Aaron keep trying to benefit the patients by finding other strategy to lower the cost for the future. So I have a question that you spend a lot of money to Jackson Lab and ask them to test the drug, and but only testing the CMAP amplitude. And why is that? Is it easier, or what does that represent?
So we tested a lot of things. I just showed you the one that looks good.
How is there like a motor function?
Well, the other thing is there are like rotor rod, there's grip strength. Just being serious, those measurements were very, very variable. Like it depends. So the CMAP was very, very consistent. So that's why I focused on that. So we're doing other ones. We're doing neurofilament, light chain measurements, other ones. But the CMAP is it's unbiased because you don't have to train a mouse to be on a rotor rod, or you don't have to have anyway. That's why.
Right. Because I saw that some data will decline when the mice age. Yeah. So when we do the experiment, also like aging is a problem. Yeah. So finding a right age of mice could be critical. Yeah. Thank you.
Yeah.
All right, sure.
And I'll keep the room open if people want to hang around and talk about other things. Hi, Richard.
One last. I know we're in overtime, but I just, again, like I said before the meeting started, what an amazing 3 years this has been. But one question, Aaron, it's a little off scope from your Ataxin 2 presentation, but you, at the very beginning, you mentioned that SOD1 and FUS patients do not have TDP43 pathology. I have been really intrigued to understand why. And maybe in Zilong's Neuro Zoom 2.0, we'll have an opportunity to consider that, unless you have an immediate answer.
Yeah, it's a really fascinating question. It could suggest that there are different subtypes of ALS that all converge on motor neuron degeneration in different ways. Yeah, I think that's one idea. We have data that I'm nervous. Whatever the word for excited, anxious to present, in which we have evidence that SOD1. Anyways, the SOD1 may be more relevant to sporadic disease than previously thought. But I'm going to present that at a big meeting next week. So I wanted to save that, but while we're just getting our ducks in a row.
Very good. Thank you so much.
So may I ask a question?
Sure, go ahead.
Aaron, this small molecule that works in the micromolar range, and I believe you said that inhibits the lysosomal ATPase. Does that do that? How much do we know about how that actually works?
So, how much do we know about how that works? So you mean its effect on Ataxin 2, or just
well,
how
no, it's what's its excuse me, how much do we know about its mechanism? Is it in this case working by inhibiting the ATPase? And if so, how's that work with regard to Ataxin 2? Can you mutate? Does it actually bind to that ATPase?
It seems like these bisphosphonates somehow mimic ATP or ADP, and they might directly inhibit the ATPase in that way. How that then leads to the cell having less Ataxin 2 is unknown, but it's an active project in the lab.
Okay, thank you.
Thanks, Ken. So yeah, I'll keep the Zoom room open if people want to chat with each other. It's great seeing everyone. If we want to see Tim Mosk in his tuxedo again.
Is tuxedo still on? Whoa, it's still on. Right.
Stay into the minaret.
Yeah, I wanna take a picture to people still stay in the room, so I can yeah, open up the camera. Let me take a group photo for the last meeting of Neuro Zoom 1.0. Yeah, let me.
I need to put on my tux. I need to put on my tux first.
Oh yeah. Let me. I'm trying to. Yeah, many. Oh yeah. Let me. Okay. You can open up your camera. Chu-Fu, I saw you.
So there will not be online
great.
Neuro Zoom anymore. There'll only be like on-site Neuro. So online Neuro Zoom is over.
All right.
Online Neuro Zoom is over, but there'll be more things to come.
Yeah. All right. Send the picture around. Use an email list. Right. Thank you all. It's a
Thank
pleasure
you,
meeting
Long.
you.
Thank you, Aaron.
Yeah.
Thanks, Aaron.
Yeah.
Thanks, everyone.
I'm sure I had many questions to ask, but I'll ask Aaron in the beer time in the ALS meeting in Shanghai.
Yeah, I hope everyone can make it to Shanghai. It's gonna be a great meeting. Oh, I've gone 3 years. So QIAGEN's been sponsoring us. I've never mentioned QIAGEN on here.
Yeah.
Oh,
that's why we can going on for 3 years.
I know.
There's strong funding for that.
Funding from QIAGEN?
Yeah. Funding for Aaron.
I've never mentioned them online until now.
Yeah.
QIAGEN.
Product placement going on.
In China, there's Tengen is like the QIAGEN kit or the kits that you use. Like, but like the secret is that Tengen is also owned by QIAGEN.
Oh.
I heard it's not as good.
Still basically the same, right? It's just
a
One of
cheaper version.
my postdocs is in Shenzhen now, and he was saying the same kit is completely different. I don't know. This is just from him. Like it's completely different results.
Oh,
from the kit that he buys from China. But I don't know.
All right, guys, I stay home for Neuro Zoom. I
Thank
need
you,
to come
Aaron.
out to work. Yeah,
great
see you,
seeing you,
everybody.
everyone.
See you.
Bye.
See
you.
See you. Bye.
Thanks, everyone. See you out there.
See you next week.
See
you in the world.
See you around.
Yeah.
Around.
I'll stick around, John.
Who's left? Everybody's leaving.
That's okay. Shinru is still here.
Shana's left. Where is she?
Yeah. The irony
Did
is,
Moscow
I don't really
leave? Moscow left. Moscow left. Need to get him back.
Yeah, it's okay. Maybe we can have like a after party or something.
Yeah.
Yeah, thanks so much, Richard, for your support every week.
It's been truly amazing. I mean, we've all said it, there's nothing to add, but the diversity of the talks, and for me, as kind of an old timer, to revisit aspects of basic neuroscience that I'm a clinician, I'm a physician, but to try to remember things that I learned 1,000 years ago. Is Zilong still on? I was going to mention something to Zilong.
I think he stepped out.
Looks like he's off. He's off. Yeah.
I'll maybe.
Yeah, I think like a lesson is like 20-minute talk, any topic, anyone can sit through that. So like I really enjoyed learning new things.
Yeah. That's been phenomenal. It really has. Okay, so we'll stay tuned for whatever comes next and in person and so on. I do have my multiple entry visa to China, but I don't know that I can make it in 1 week. So
no, that one's probably canceled. So you have to like
they canceled all those visas. They canceled all those visas there.
All right. But anyway, great to meet, to have this moment to talk and thank you guys again. Okay. Bye-bye.
Thanks so much. All right, John, I think I'm gonna drop out.
All right. I'll talk to you later. Let me know. We'll talk about